Yeah. So basically, what this paper encapsulates is a technique for cell programming. So programming the entire genome of the cell using a highly efficient technique called MAGE. So I'm going to come to what MAGE is a bit couple slides later. Yeah. Yeah. So what was the motivation for developing maize and why did it start getting used? So basically, natural evolution produces enormous genetic and phenotypic variation in the sense that all the organisms that we see today they've been all produced with genetic variation and mutations, right? And variants that are fit for a certain set of circumstances or situations always have an inherent advantage, which basically is the basis for natural selection. But if we try to do the same thing in vitro, it is extremely time taking, and the evolution of organisms to generate genetic variants is not in practical lab time scales. It takes a lot of time, and the amount of time you take to generate like genetic variants at the scale of what nature does is very long. So there are methods of directed evolution in organisms to produce an artificial phenotype. But again, firstly, it is very laborious to do so. And secondly, it's only one gene that can be diversified at once and not multiple genes or entire genetic networks fall apart. So the first solution that, the first in vivo solution which came about for doing this was called recombinating. Recombinating is basically an in vivo method to generate genetic diversity using single-stranded DNA and the beta protein from the lambda phase red system, which came to be known, which is basically a system of allelic replacement. So what happens in recombinating is we introduce single-stranded DNA oligonucleotides being nucleotides of short lengths, which integrates with the chromosomal DNA through the Okazaki fragments formed during replication. And then these single-stranded DNA oligonucleotides are annealed to the lagging strand by the beta protein, causing generation of modifications in the host chromosomal DNA. But the thing was this method was very interesting, but when it was like sort of put into paper, it had only been performed on a single gene scale, whereas the system could be realized to have a much larger magnitude of implementation to diversify entire genome simultaneously. So basically, the people who wrote the paper to illustrate recombinating weren't aware that this could be used in a much larger scale. They had basically showed it to work on a single gene scale. Now, so going to the bit into the specifics of how this recombinating or specific oligo-mediated allelic replacement works is. It's been characterized by various factors affecting efficiency and extent of replacement. Firstly, the efficiency search mismatch mutations is correlated to the sequence homology. So basically, the similarity between the between the sequences of the oligonucleotide and the target chromosomal sequence. But on the other hand, for deleterious mutations, it's correlated to the size of the deletion. So basically, these are characterizations of how oligo-mediated allelic replacement work, and these were taken into account while designing the MAGE system. Okay. Secondly, the two-state hybridization free energy, that is the free energy associated with the oligonucleotide sequence and the host target sequence, determines the efficiency of allelic replacement, which in turn is determined again by the homology, like the similarity between the sequences. Hence, let's say if you have a pool of degenerate oligonucleotides, so what a degenerate oligonucleotide is basically a set of a specific length of nucleotides, each nucleotide which can have a possible set of bases. So that these are represented by letters such as N, R, P. So N basically means that position can have all the four bases there. R means it can have only A and G bases there, and so on. So if we have a pool of degenerate oligonucleotides, more the homology of the oligonucleotide with the target sequence, so like more the similarity of the sequence of the oligonucleotide with the target sequence, essentially more the hybridization free energy, which causes a higher frequency of that oligo getting integrated into the genome, forming something called heteroduplex DNA. So if you look at the graph, this is a general scheme as to how MAGE works. We'll come to this later. Firstly, how in this one, characterization of allelic replacement efficiency. We can see that as for mismatch and insertions, as what do you say, the size sort of increases, the efficiency decreases. Same, but for deletions, it decreases much slower, so it's correlated more. Most importantly, the hybridization free energy can be correlated best with the efficiency. Greater than energy in the negative direction, more the efficiency of replacement of alleles. So what these guys propose is something called MAGE, multiplex automated genome engineering. It is a highly efficient automated method to simultaneously modify multiple locations on the genome on a diverse range of length scales. So basically, this solution sort of modifies genome on a long range of scales. So it can span from a single nucleotide. So you can basically modify or diversify one nucleotide, and that can go on to entire genes. So entire genes, coding proteins can be modified using this system. It basically involves performing what we saw in the past two slides, allelic replacement on multiple places of the genome using oligonucleotides of highly specific or highly degenerate oligonucleotides. Sorry, that was I meant that only once. Steps involve so basically how this main system works schematically is the colonies with whatever phenotype we want is cultured. And also the beta protein of the lambda plate system is also expressed within these colonies. Right? Electroporation based transformation of the bacteria with the oligonucleotide. So the bacteria transformed with the oligonucleotides, and then general washing, and if they are preserved, resuspension of the bacteria back to the culturing phase. So this all is done in cyclic manner, and so each cycle is called a MAGE cycle in this presentation. So yeah, so this is the time what a MAGE cycle looks like. First you grow the cells, then you induce the beta protein expression from the lambda phage system. Then you, if you want to preserve them, then you chill them and then again resuspend them when you want to transform. Add the oligonucleotides to the solution, deliver them to the cells by electroporation, and then you then after selection of the cells, which is not exactly given here, selection of the cells showing the phenotype, you then recover the cell, proceed to the next cycle. And this is how sort of the MAGE automated schematic diagram for MAGE looks like. So you have various pumps and you have storage units to keep the cells. Right. So first, determination of efficiency of MAGE. This is the first experiment that is elucidated in the paper in order to like sort of determine how efficient MAGE is as a system. So what for this experiment, three different types of 90 so basically 90 nucleotide long oligonucleotides were used to target LAG-C gene in different populations of the ECNR2 strain of E. coli. So the three types of oligonucleotides used CN6. So basically, consecutive N6 bases. N means, like I said, N can have any of the four bases in them. Again, CN30 means consecutive N13. So 30 degenerate bases. CN6 is 6 degenerate bases. And IN6 is interspersed N6. So that means basically 6 degenerate bases interspersed between 30 in general bases. So these three oligonucleotides were used, and the LAG-Z region was sequenced in 96 different clonal isolates. So after culturing whatever the clones of one, they were isolated, and these were used to check the phenotypic variation in the population after 2, 5, 10, and 15 MAGE cycles each. So after every of these number of cycles, the region was LAG-Z region was checked and sequenced, check diversification. So what did it result in? Successive cycles of MAGE caused drastic divergence of LAG-Z from the wild type. For the oligonucleotide CN6, there was a 3.1 base pair change per cell. So this quantifies to about 4.3 billion total changes in the loci of all the cells. And although the cell population at any point in time denotes a subset of all the possible theoretical number of variants, the paper said that more variants can be generated out of the more variants, and the original size of the cell population can be generated through using more successive MAGE cycles. And given the cyclic and scalable nature of their approach, an integrated prototype device was constructed that automates the maze process to enable fast and reliable cellular programming. So basically, what they're saying is that the maze process is highly scalable and also it's cyclical, so it's easy to implement. So this diagram basically sort of shows the frequency of base pair change in the population with respect to the three nucleotides. So you can see that the N30 nucleotide has a large base. It only shows a 7+ base pair change, even though not much in frequency. While you can see that the CN6 oligos being since it's smaller in size, you can see a large distribution of the change in base pair as you increase the number of base cycles. So in the end, after 15 cycles, we can see that there is a huge change in the almost all of the population has a change in their genome. So this is how efficient MAGE really is. Right. Now the second experiment which elucidates is application of MAGE, like how MAGE can be applied situation in order to do so. So today we learned about the DXP biosynthesis pathway, and so they use the same experiment to actually show the application of MAGE. They basically use MAGE for optimization of a genetic anatomy rather than simple diversification of a gene. They use the DXP biosynthesis pathway to overcome the selective isoprenoid ligogames. They use a strain ECHW2. It's basically why it contains this plasmid that is necessary for the final steps of lipopene production. There are 20 endogenous genes, each documented for increasing lipopene yield, which were targeted to optimize translations. Again, specifically 91 oligonucleotides containing degenerate RBS sequences. So again, like I said, degenerate means they can have any of the bases in this consensus sequence. So D implies both A and G can be there. All three A, G, and D can be there, and R implies A and G can be there. So these generate a number of possible sequences, and these are flanked by homologous sequences on each side. And the replaced RBS regions were designed to be more similar to the canonical SD sequences, hence enhancing translation efficiency. Additionally, other than the 20 endogenous genes which were used for increasing lipopene yield, 4 genes from secondary pathways, which themselves cause generation of nonsense mutations. So nonsense mutations basically produce stop codons inside the ORF, and so in order to stop them from doing so, they were inactivated. And therefore, total 20 plus 4 genes were modified to maximize lipopene production. Right. So you have 24 genomic targets, out of which 20 are optimized for translation efficiency, and 4 are gene knockouts, so that they don't cause production of stop codons in the middle of the ORF. And these are the genes that basically code for the biosynthesis pathway in the strain. So we'll come back to the graph later before we should go to the result. Yeah. So results of experiment 2. More or less 15 billion distinct variants were generated. That's a very huge number. The colonies showing intense red pigmentation on the LB plate were selected for each cycle. Variants from about 10^5 colonies were isolated after 3 to 35 mate cycles. So ranging from 3 to 35 mate cycles. Some of which exhibited a 5-fold increase in lycopene production as compared to the ancestral ECWH2. So that strain was originally used to run through all the mate cycles. And sequencing of 6 variants, 6 different variants, 6 of any of the 15 billion different variants, revealed the convergence of the RBS sequences to a consensus-like SD sequence in genes located at the beginning and end of the biosynthesis pathway, as well as various gene knockouts from the secondary pathways, which basically says that the RBS sequences do converge to the most optimized genes for lipopene production. Secondly, tuning parameters can be assessed in isolated cells. So, which optimization of which gene, along with which gene, causes the maximum lipopene production can also be assessed in isolated cells. For example, the IDI gene, which was one of the enzymes in this IDI gene. Yeah. In this variant, ECHW2A, the IDI gene, only the IDI gene was optimized, which caused a 40% increase in production, while the optimization of both DXS and IDI genes caused an increase by 390%. That's in ECWH2E variant. Another optimization at RPOS and DXR, as well as inactivation of YTJC. YTJC is one of those part of the secondary pathways which was inhibited to stop nonsense mutations, which this causes the growth rate, improve the growth rate of the respective variant rather than actually causing an increase in lycopene production. The improved growth rate implies an increase in lycopene production. But yeah. A point of interest here is that the RPOS is actually an automated polymer signal, which is the master stress response regulator. It's up to the optimization of RPOS. It significantly increase the cell's tolerance to resistance, tolerance to stress. And since lycopene is a highly hydrophobic molecule, it increases the cell's resistance to accumulation of lycopene inside the cell and proceed to grow normally, even though there's a high lycopene in the cell. Before we move further, let me show this graph. Yeah. So we can see that as lycopene, the correlation between lycopene production and main cycle strains, as more and more genes are optimized and the others, like the secondary pathway genes are like inactivated, we can see that lycopene production increases to about 9,000 ppm, which is basically zero in the ancestral strain. So you can see that the amount of jump in like production that can happen only by optimization of the biosynthesis pathway. So concluding, the diversity generated by male is highly adjustable and the specificity of target is also high. So basically, when we need to generate a high variation in sequences, we use highly degenerate oligonucleotides. While if we need to specifically engineer some sequence inside the genome, then we use highly specific oligonucleotides. And they use a similar thing here. They used well-defined oligonucleotides for specifically knocking out the secondary pathway genes, the four colored ones. While the high diversity generating oligos, which are highly degenerate ones, were used to optimize the RBS sites. So this way you can sort of optimize between specific and degenerate. I mean, also the percentage of specificity in each oligonucleotide in order to completely optimize the pathway of any degradation of any sort. It has also been used to perform whole genome decoding, to enhance incorporation of non-natural amino acids and construct safe multi-phase resistant strains. Finally, it's an accelerated evolution platform that permits repeated introduction and maintenance of many neutral or deleterious mutations. So basically, we are using this technology, one could engineer cells with higher transfection efficiency, which basically is making cells that are naturally competent, in order to engineer them better. Increased allelic replacement efficiency, which is what basically recombinating is, making them more susceptible to recombinating, and perform large-scale artificial chromosome engineering. So basically, on a chromosomal level, if you want to change and optimize on the sort of pathway, then you can do this. And financially, this entire experiment had not this experiment, but this was quantified that if we use only $30 worth of oligonucleotides for this kind of experiment, we can generate a 27 base pair modification per cell at full degeneracy. Which is, mind you, we already had a 3.1 base pairs change in this experiment that caused a 4 billion modifications in the entire population. So imagine what a 27 base pair modification can do, and this is only $30. So basically, what I'm trying to say is it is quite economically viable, and development of pipelines in order to organism to optimize some pathway or multiple pathways is very easy, financially speaking. So this can be used for integration of hardware, software, and hardware to engineer and evolve plant animal systems. And this in general can make greatly benefit ecosystems, and we can engineer various types of cells in order to improve the state of our ecosystems right now. Yeah, thank you.
Have a super big audience, but still the presentation deserves an applause. Okay, any critical assessment?
This they have they have quantified it some bit, but they haven't quantified a lot as to what the allelic replacement efficiency how it's working, and also they haven't gone into like the depth, the mechanistic depths of how multiple mediated allelic replacement is works. So they have just it's a very functional approach they have taken. They have just taken an input, they wanted to do something, and they just ran it through a main cycle. I mean, I get that it's sort of the point, but just purposes they would have gone deeper and checked out mechanistically how it works.